Thank you.
Thanks, Hi. Well, to start, I guess, the first thing that comes to mind is, how does sequencing fit with policy? It is a BVD, so that's a start. But what I want to talk to you today is not about the sequencing per se, but what it tells us and what can be done with it, especially in the policy context. Try that button. Yay! So the first thing I want to do really is to thank the sample providers who, since about 2013, have donated more than 5,000 serum samples from BVD PI animals, mostly from Scotland, but from other parts of the UK as well. And we've got those mainly from SAC Consulting, the SRUC Diagnostic Lab, and also from BioBest. So I'd like to show our appreciation for both the samples and for the information we get alongside them. So what I'd like to talk about today is an example. There's another example on the poster that goes with this talk, but this is really just an analysis of the herds in our biobank which have the largest number of calves born on that premises. And we got that information using the first 6 digits of the UK numbers for those animals. So we found there were over 40 herds into which were born more than 10 PI cattle in the space where we have data, which is about 2014 to 16. We looked at about a dozen of those samples, a dozen of those herds, and that amounted to some 200 bits of sequence data. And looking at the patterns there, we distilled it down to look at actually 2 premises as an example. Now, one of these premises had about 11 PI cattle. It's the blue squares on the diagram on the right-hand side. And those animals, when we sequenced the BVD in each of those samples, were identical. And that's why there are a straight line of little blue symbols there. So they're all identical sequences. We take from that this farm, when the dams of these calves were infected during pregnancy, they were infected from a single source, and presumably from a single PI animal, since the animals in this particular farm were born over at least a year. So that's a persistent source of infection for those pregnant animals. At the other extreme of this, we have the premises illustrated by the red triangles on the tree. And what we see there is a similar process whereby the majority of animals that they live in on the right-hand side, again, all had the same sequence. So these are, again, representative of a single strain of BVD, a single source of infection for the dams of these animals. However, in addition to those 11 animals with identical sequences, we saw that there were another 4, and they weren't just a little bit different, which would show up as a tiny tick away from that horizontal line. They're actually quite significantly different. So they differ by more than you'd say these are the same strain. So what we've got on this one premises is the majority of infection of the dams came from a single source, but in addition to that, there were small amounts of infection taking place at different times from another 4 distinct BVD strains from different sources. Now, obviously, we can't say what those sources actually were, whether they were contaminated equipment, boots, individuals, transport, or whether there were other PI animals brought onto that farm at different times. What we can do, and I guess what we can do with, if you like, additional data, is we can look at, in addition to the sequence data, we can combine that with movement data, and that's been talked about already, and it'll probably be talked about later as well. By incorporating movement data into that, we can ask the question of not only where were these animals, because we know where all these animals were born, where were the dams, where were other animals on the farms, where are there other PIs that moved onto this farm from somewhere else in the appropriate time frame. So that's my message for today. Sequence tells you so much, but you'll always need more data. Thank you.